One of the, well, and as I think you're all aware, one of the major aspects of MS is a loss of myelin, which is the protective insulating coat around axons. And this not only causes an immediate loss of conduction in those axons, it also leaves the neurons vulnerable to irreversible damage. So actually getting this damage repaired as soon as possible is really in the best interests. Also, in the course of MS, initially people tend to repair this demyelination, and this process of repair is called remyelination, reasonably efficiently. But as the disease progresses, this process of repair tends to become less and less efficient, and this probably is one of the contributing factors to the progression of the disease. So my primary interest is actually understanding the signals that either promote or block the myelination process. And these signals can really take multiple forms. So myelination is, in the central nervous system, myelin is formed by oligodendrocytes. And these oligodendrocytes actually arise from another cell type that we mostly just refer to as oligodendrocyte progenitors. So any signal that encourages the generation of oligodendrocytes from these oligodendrocyte progenitors should, in theory, encourage remyelination. Conversely, any signal that blocks the production of oligodendrocytes will, in turn, block myelination. And in addition to that, there's also signals provided by the neurons that actually block or allow the oligodendrocytes to myelinate them. So the work I was performing at Stanford was to identify a gene that's actually expressed only by mature myelinating oligodendrocytes within the brain. And this really gave us a clue that this gene may have some important role in myelination. Usually if a gene is specifically expressed by a given cell type, that gives some indication of what its role might be. And this gene was actually encoded a transcription factor, and these transcription factors are genes that turn on or off vast cohorts of other genes. So you can really see them as the conductor in a symphony, sort of orchestrating everything that goes on around them. And effectively what I was able to show using a genetically modified mouse is that if you deleted this genome from this gene or transcription factor from the mouse, the mice were unable to generate myelin at all. There was basically no central nervous system myelin in the developing mouse. And perhaps more importantly, I was able to show that if you take this gene and force the oligodendrocyte progenitors to express it, they actually will push towards becoming oligodendrocytes and expressing a series of genes that are normally associated with myelination. So based on this, we were really able to show that this gene is an instrumental part of a myelination process. And work that I'm conducting now at Melbourne University and the Howard Florey Institutes is taking this gene and trying to look at its role in myelin repair in the adult central nervous system. So work I've been conducting over the last several months has shown that this gene is actually turned on in the context of successful remyelination. And I'm now in the process of using mouse genetic models to either inhibit its expression or increase its expression to see what effect this has on the efficiency of remyelination or myelin repair in the mouse.